Very cool. All right, well, let's get going. First of all, thanks for attending today. My name's Elliot Swartz. I'm the principal scientist for cultivated meat at GFI, and I'm joined today by Bert Frohlich, owner of BioFarm Designs, who led this work that we'll be presenting on cell growth modeling for cultivated meat production. Today also marks the release of the full report, which we will drop the information to download that in the chat, or you can also scan this QR code. I'll mention that the report is fairly long. It comes in at about 150 pages, so it's more of a mini textbook. And so alongside of this, we've produced a technical summary that summarizes the key points of the report. And it's also important to know that all of the sort of data spreadsheets and workbooks are available for download as well. So with that said, we won't be able to cover all of the content of the report in today's webinar, but we hope to really be able to give you a sense of the content so that you can go and read the full report for all the full details. It is definitely worth reading if you are working in cultivated meat or process development. There's a lot of new analysis and insights that I think you'll definitely walk away learning something new and be able to take something forward into your work. So we'll present the information. I'll first provide some context and background for why we decided to embark on this project. And then I'll turn it over to Bert, who will outline the modeling framework and approach. We'll discuss section 3 of the report, which is really the bulk of the report around the data, the parameter data and data gaps. And I'll end with some conclusions, recommendations, and future directions. And we should have plenty of time for Q&A at the end. So before we begin, for those unfamiliar, GFI is a 501c3 nonprofit. We're a science-driven think tank helping to build a more sustainable, secure, and just protein supply. Across our areas of science and technology, corporate engagement, and policy, we're really trying to create an innovation ecosystem that will advance the science and adoption of alternative proteins. We have teams based all over the world in the United States, Brazil, Europe, Israel, India, Asia Pacific, based out of Singapore, and also Japan. We all operate under the same mission, and our work is powered entirely by philanthropic support. And so if you like and find value in the resources that we produce, please do consider supporting us. So let's talk about why we decided to embark on this project in the first place. And it really goes back to the overarching question that pretty much everyone has, which is what is the cost of cultivated meat production? And to answer this, what has happened over the years is a series of techno-economic analyses, or TEA studies, starting in 2021 by David Humbird, also work by CE Delft that we funded, and a group at UC Davis. In the following years, other academic groups and more recently, even companies have began to publish their own versions of TEAs, in some cases using their own data, which is great and has provided sort of new insights into the cost drivers of production. However, we will discuss, some of the limitations with these existing models, and in our report, really map out a future where we can hopefully work under an improved modeling framework, incorporate real data from relevant cells and processes, and be able to really leverage these models in new and more predictive and useful ways. And so we'll be talking about all of that today. So while the existing sort of techno-economic analyses have been very useful in identifying the cost drivers of cultivated meat production, the media, the bioreactors, the overall infrastructure, they've kind of been overinterpreted as conclusions about the techno-economic viability of cultivated meat. And the truth is that, existing models do have several limitations. Most of them evaluate just, a limited set of a predefined process. And so there's a lot of different bioreactor types being used in the industry, a lot of different modes of operation that are possible, scales of operation. A lot of these just haven't simply been analyzed. They also simplify complexity of cell growth during, the bioreactor culture via a series of assumptions. And so really the existing models are not necessarily, mathematical equations. They're really more of a series of assumptions. And then finally, they rely heavily on data taken from cells used in other industries for other purposes, like the biopharmaceutical industry, where the cell isn't really the product. And so there's always this question mark of, how relevant are those data? And so, as we'll discuss, we want to be able to move towards models that can really enable one to discover the true potential performance envelope of real dynamic cell growth under various process conditions, and be able to really make predictions and make test hypotheses around process changes that you might make to overcome challenges that are known when it comes to scale up. And so this is a general sort of high-level overview of the study approach. We're going to walk through each of these, starting with the modeling framework, and I will turn it over to Bert to talk about that.
Great. Thank you, Elliot. Great to be here. Thanks for joining, everybody. So really, the first question is, what does this model need to do for us? And we put together this list of specifications, if you will, and there's probably as many models as there are applications out there, but we want to be very specific about a model that can help support future TEAs. And by extension, that means also looking at the bioreactor and being able to assess its performance, because that is a significant part of the economics for cultivated meat. So we want to be able to predict the volumetric productivity, and that has a very special key performance indicator. We can talk more about it, but it has the units of grams cell mass produced per unit time per unit reactor volume. So keep that one in your head. Secondly, we want to be able to optimize feeding strategies to the reactor, because as you'll see, the way nutrients are fed to the reactor can affect the performance of the bioreactor. Thirdly, we want to be able to evaluate the effects of heterogeneous conditions on the overall performance, because very likely the scales, bioreactor scales that are going to be required for cultivated meat are going to be enormous. And my little picture to the right is my attempt to show how a cell, that's the dotted line, might move through a bioreactor. And it's going to be exposed to many different microenvironments, and those could be, changes or gradients in shear, oxygen, nutrients, pH, and even temperature. And that situation is only going to get more complex as the viscosity goes up when we achieve higher and higher cell concentrations. Fourthly, we also want to be able to improve these bioprocesses, so all three aspects above. I want to be able to look at trade-offs between physical and biological conditions, and specifically the effects of temperature and pH. And we'll say a little bit more about that in a bit, but I think those are opportunities for overall process improvement. Number 5 and 6 are perhaps a little bit more indirect, but we at least want to use a model as to do what-if scenarios and look at the effect of various cellular adaptation strategies, alternative substrates, and altered biochemical pathways. That's not to say that this model would be used for designing cell lines and media. Those are probably different models. We can say that something about that briefly. But anyway, that's it. And next slide. Because TEAs are all about economics, we have defined this optimization parameter that we call performance to cost ratio. And you can define the PCR for any level in an operation. On the left side here, this picture denotes for an entire facility, where you look at the total output from that facility compared to the total number of dollars in, and that would include the capital requirement as well as raw materials. So essentially, the PCR is an inverse COGS, right, in terms of kilogram per dollar. Or in a more simple, on the right, the PCR could be defined just looking at the production bioreactor, where we're looking at the amount of cell mass that can be produced compared to the price that we're paying for that bioreactor. So there's a full spectrum there. And yeah, that's right, Elliot, thanks. To achieve those specifications, we're proposing this model structure shown here, and it has two main components. On the right is the cost model component, and you can see we have many different layers there that you can then assess for a given operation. And that is not the subject of this work. The long-term vision is that would be a cost database that would be able to estimate prices for equipment and processes. And that envision there that GFI has is that could be an open source tool in the future, but that's for another day. our focus here is on the performance model, which is on the left, and you see these three uh, three layers to that. And in the next slide, we kind of peel that apart. So the focus of this work and this paper is really around the cell growth model. But we just wanted to put this in context for you. Surrounded by that would be the bioreactor environment model, which is going to simulate the physical space in 3 dimensions. And that most likely requires some sort of computational fluid dynamics. And then surrounding that would be what we call the bioreactor system model, which would then dictate how is the reactor being operated, what are the control functions, how are you feeding substrates, so on and so forth. So on the next slide, the reason that we showed the cell model inside the other 2 layers is because the cell model was going to have to be executed. This is from the modeling sense, many, many times. So if you have a computational fluid dynamic model of the reactor, which is in the middle, that represents the bioreactor environment. For every fluid element and for every time increment, you'd have to run the cell model if there's in fact a different environment in the reactor. So it's going to be computationally quite complex. And so we want a model that is fairly simple, that we can use, and that is still predictive. So the big question is, how complex does it need to be? On the left here is a picture of probably what is minimally required. We know from the characteristics of animal cells that we've listed over to the right that they use glucose and glutamine as their primary substrates, and they use oxygen for aerobic respiration. They have the primary metabolites that are generated from the consumption of those substrates is ammonium and lactate and carbon dioxide. And excuse me, if the pH drifts, we often add base to a reactor to control the pH, and therefore the osmolality also goes up. So that's kind of the minimum. But depending on what the data tells us, we may need to include more to get it fully predictive, something like pictorially shown on the right. So to put this into biochemical engineering jargon, we included this diagram in the paper. The diagram that's included is the one that's in the lower middle there. But to that, I've added, just to put this in perspective, and the axes on that diagram is, if you can't quite read it, but on the x-axis, increasing model complexity, and the y-axis is increasing computational intensity. And even though some of the more complex biological models that are used in the biochemical engineering field, segregated and structured models, are still fairly simple compared to things like metabolic flux analyses, genome scale models, which have recently started to be published. And it's not the subject of this, but we just wanted to point out that those are out there and certainly have use. But if you try to couple one of those with a CFD model, which are the boxes to the left, you'd have a very complex set of relationships equations and probably would not be able to run that on a normal computer. Now that may change someday, but for the time being, what we want to do is we want to couple something that's simple as possible on the biological side with something that's manageable on the bioreactor environment side. So at this point, I think turn it back to you, right, Elliot?
Yes, let me hop back in here. All right, so we'll switch off here and talk about, now that you have a sense of the sort of overall conceptual framework in which we're operating in, we'll now jump to really the majority of the work, which focused on gathering and evaluating the parameter data that would be used to fit, and then fitting the data to different types of models and understanding whether data are currently available and sufficient, or whether significant gaps exist. And as you can, perhaps guess, that with a new field, there are significant gaps with respect to data, as we call out in the report. So we'll walk through some of the major sort of areas in which we explored, starting with questions around cell size, mass, and composition. And going back to what Bert said about bioreactor volumetric productivity, really knowing the cell mass is going to be essential in order to predict process productivity. And so when you look at the literature, you actually find that most cells, single cells in suspension, tend to be spherical, and they also tend to be about 10 to 20 microns in diameter. Exceptions to that might be fat cells that can be up to 10 times larger, but nevertheless, across existing cell types that have been analyzed, this is generally what you find. Now, the problem is that a lot of people take the diameter of a cell and then back calculate the mass. And this is actually, as we show in the paper, quite a poor proxy for its mass and its compositional makeup. If you look at actual cultivated meat, TEA and LCA models here in green, and the assumptions that they use for single cell size or single cell masses, the dry cell weight is generally assumed to be around 900 to 1,000 picograms, and the wet cell weight between 3,000 and 3,500 picograms. But if you look at the literature and studies that evaluate actual measurements of cell mass, these are overestimated by a factor of about 2 or 3. And so it's very clear that we need actual quantitative measurements of dry cell weight for cultivated meat relevant cells, and these really need to be quantified themselves rather than attempting to estimate it from other sort of parameters. And one of the reasons for this is because even if you look at real world products that have been approved in the marketplace, the water content is highly variable. And so if you're doing back calculations, you're just sort of adding a layer of error that's possible into these mass measurements that are really critical foundational values that will go into these models. One of the other interesting observations by looking at the literature is that, if you look at the mass and volume of a cell over the span of typical growth rates that it will experience in a cell culture, you can see that the volume and the mass can change by more than a factor of 2 during this time. Not only does the mass change, but also the biomass composition will change. So nucleotide and protein content, for instance, can vary based on the cell cycle and also the overall phase of the culture, whether it's in exponential or stationary phase, for example. And actually, not a lot of emphasis has been placed on this in other industries because it's perhaps less important. But with cultivated meat, it can really influence the biomass composition, which really turns into the nutritional content of the end product. And so discovering more about how to optimally harvest your cells is going to be also important, not only for productivity, but also the ultimate value of your end product, which might be tied to, let's say, the fat content or protein content of the cells. So some of the takeaways from this subsection of the report, as I mentioned, we really need more direct measurements, not only of wet cell weight, but especially dry cell weight, so that we can't stop relying on cell size to make assumptions. We also need standardized protocols for actually taking these measurements. The way that you measure a wet cell weight might vary between labs, and so having standardized methods are going to be important, so we're speaking the same language. As I mentioned, more research needed on how growth phase and cell cycle affect mass and composition. Also not mentioned here, but in the report we talk about and show data around differentiation, which is going to also be quite important. And then finally, the data really should be collected in defined media across the relevant cell types. This will allow us to move away from some of the proxy measurements that we currently take from cells used in the pharmaceutical sector, for instance. All right, I'll turn it back over to Bert to jump into more of the specific details.
Okay. Yeah, before moving on to how we go about modeling growth, I want to introduce a few key terms and variables that we'll be using throughout. First and foremost is the specific growth rate of a cell, and mu, and it's typically referred to. And I know that a lot of you are used to thinking about cell growth in terms of doubling time, but from the mathematical perspective, that just kind of confuses things because it's an inverse relationship. But specific growth rate we use routinely. If you want to think about what it actually means, you could visualize that as how many cells can a given cell produce per unit time. And the cells cancel their inverse days. The same can be done with mass, and we'll be using mass primarily for the reasons Elliot talked about. And you can visualize that as how much cell mass can be produced per unit cell mass per unit time. So same units, but I think it's important to point out that the number or the growth rate calculated based on cell numbers not always the same as that based on mass. And as an example, you can visualize cells differentiating, kind of brings it home that they're gaining in mass, but not in number, right? So those are not the same. It's important to keep in mind. Then we have the variables. We have X, which is typically used for cell concentration, and we typically refer to that with mass units here. S for substrate concentrations, could be glucose, glutamine, oxygen. Inhibitor concentrations, I, uh, lactate, ammonium, and we'll talk more about those. And then finally, we have the specific consumption and production rates, which we refer to typically as Q sub S and Q sub I. And you'll notice there that we are standardizing on units of millimole of substrate or inhibitor per gram dry cell weight per day. So we're normalizing all of these to the dry cell weight of the cell. Now for feed conversion ratios, we do state those in the report. Those are a little easier to visualize in terms of grams per gram, and we'll report some of those. So moving on to modeling growth rate, what's typically done is for a given cell line, there's a maximum specific growth rate that's characteristic of that cell line, and that's kind of used as the cap or the maximal growth rate per definition. But it's also important to note that that mu max is also a function of temperature and pH. And over on the right, there's some of the data. We couldn't find much, but there's some data we could find that expressed growth rate as a function of temperature and pH. And you can see that they have relatively sharp optima. And you may ask, well, why do we care? Why not just operate the bioreactor at the peak value? This is optimal for the given cell line. But we just want to point out that temperature and pH also have profound impact on some of the physical characteristics or parameters in the bioreactor, such as gas solubility, viscosity, other things that could also influence performance of the reactor. So we think that this is an area of research we pointed out as a gap, that there's very little data available on the effects of temperature and pH, and something that should be pursued. So modeling growth rate, the actual growth rate from the maximum, is what's typically done is we apply these factors, each of them a value between 0 and 1, times the maximum growth rate that account for these various phenomena. It's the NFG is a nutrient factor for growth, and it's typically broken up by component or substrate. So there's a separate factor for each of the substrates. And below that, I'm just showing some example kinetic profiles that are typically presented in the literature, biochemical engineering literature. The most common one being the Monod kinetics, which is the model number 1 shown there, that saturates at a certain, beyond a certain concentration. Then we have an inhibition growth factor. A similar treatment, or each inhibitor is assigned a factor between 0 and 1, and those models are typically fit to one of the curves below that are also published in the literature, the most common one also being number 1, which is referred to as an inverse Monod relationship. And then finally, the shear factor. We are not going to go into that in this work. It's a very complicated field. And suffice it to say that it's a big gap for us in terms of understanding the effect of hydrodynamic shear of various types on animal cells. Even for conventional cell lines using BioFarm, there's not a whole lot of quantitative data on that. So that's a whole nother area of research that needs attention, because they're sure to experience that in a large-scale bioreactor. Okay. So let's take these one by one. This would be the first term, the nutrition growth factor. We looked for data for the 3 major natural substrates of animal cells. And we found quite a bit of data for glucose and glutamine. And the 2 graphs on the right are just examples. Those are probably the cleanest data sets we could find, just to show you as an example. The green curves are growth rate versus the glucose concentration. And we can extract the Monod constant, which is the K sub S in the equation shown there, for each of those substrates. And when we looked at that for all the cell lines that we could find, and we tabulated these values in the report, they covered quite a range. And you can see there the case of glucose, it ranged from 0.02 to 5.7 millimolar glucose, which is a very wide range. It's almost 2 orders of magnitude or more. And so that's quite a range. We did see a loose correlation with those studies that use serum in the medium and those without. And we saw the same situation for glutamine. Again, variable and some separation, but we can't really say that those are statistically significant observations because there's so few data available in serum-free media that we really couldn't say that for sure. But it's certainly reasonable to think that serum may have an effect on cells in terms of enabling them to take up substrates at lower concentrations. So that's a gap, and there's some more information required there. Just as a note, we also looked at death rate, and those are also on the graph, but we won't have time to get into that. But it has a similar treatment mathematically. Then next, oxygen. Yes, we also looked at oxygen. Not as much data available for that, but fortunately there was one exercise by Miller et al. That was done very cleanly with a complete data set, and we're able to look at that. I point this out because it's an interesting profile. It has a very sharp optimum of growth rate, or mu max occurs only over a very narrow range of oxygen concentrations, and then it goes down. And what that means is that oxygen is actually toxic to cells at higher concentrations, even below 100% of air saturation. So to describe that behavior, we added a substrate inhibition factor, which you can, Elliot's pointing to, which consists of two parameters, IC50 and an exponent. So between that and a monad model on the left, you put those two together, you get a parameter model, which actually fits the data quite well. It's interesting to point out that the K sub S is much lower than it is for glucose and glutamine, which points to the fact that animal cells are very efficient at picking up oxygen at low concentrations, which makes a lot of sense. But again, very little data for oxygen in general, and more needs to be collected for cultivated meat relevant cell lines. So on the inhibition side, we have the IFG. On the right is an example of all the data we collected for ammonium. We did the same for lactate, CO2, and osmolality. And we fit those data sets to various different of the models that I had pointed out previously, and we found the one to the left here, which has a double exponent, actually fit the data the best. And the purple line, which is the mouse hybridoma, we fit that particular equation to, and those are the three parameters, and fits it quite well. So these empirical models do appear to explain, or at least model that behavior, but we want to make you aware that there's a big caveat here, and that is in the literature there was several instances that was reported that that cells are able to adapt. And the way most of these studies have been done is to add a bolus addition of ammonium chloride, in this case, at various different concentrations to an actively growing culture. And when you do that very suddenly, the cells don't have any time to adapt, and you get high inhibition, and you even get cell death. So I think the way these studies were done is not truly representative of what's actually going to happen in a bioreactor under conditions of prolonged exposure. And it also points out some opportunities for adaptation. So we just want to make sure that's clear. So we would need better data to represent inhibition that is more appropriate for actually what happens in a bioreactor. Okay, enough said on that one. Let's move on to substrate consumption and metabolite production. This is a model on the left, is the PERT model, which has been frequently used in the biochemical engineering literature to describe substrate consumption. It was originally proposed for bacteria that were growing on a single carbon substrate. And you have two terms there. On the left is the growth associated term, which you can see is associated with the growth rate mu, and a second term that is non-growth associated, which is related or referred to as the maintenance requirement, the maintenance term. And if you plot that equation on coordinates, growth rate versus specific consumption of the substrate, you should get a straight line if this model holds. And the slope of that line would be the equivalent of the inverse biomass yield, which is YX sub glucose. That would be, for instance, grams dry cell weight per gram of glucose consumed. And the y-intercept of that would be the maintenance term. So if we can predict the consumption of the substrates, then we should also be able to predict the production of the metabolites or the inhibitors. So on the right side is a hypothetical example. If there is a constant yield factor, in this case, for lactate on glucose, if you plot the consumption rate of glucose against specific lactate production, then you should get a straight line there as well, where the slope is equal to the yield coefficient. And for lactate, that slope would be about 2 as a theoretical maximum, because there's 2 lactate molecules produced per glucose consumed. So that's the theory anyway. The next slide, we took that PERT model and we applied it to glucose in this particular case. We did the same for glutamine. There was almost no data available for oxygen, so it didn't really bother. But you can see on the panel on the right is all the data that we could find for glucose consumption by animal cells, many different cell lines. But the data is quite scattered, as you can see. The slopes are similar, but not the same. There's some variability there. But more importantly, there's little to no maintenance coefficient. So the y-intercept of most of these plots goes through 0, which is strange because glucose, we know, is an energy substrate. But nonetheless, we tabulated the slopes for that, and essentially the slope is equivalent to the inverse yield and apparent yield because the maintenance term is 0. And when you convert that to feed conversion ratio, we had quite a range. And that number ranged from 1.6 to 3.6 grams glucose per gram dry cell weight produced. If you ask me, that's a pretty lousy conversion. It's almost 3 times as much mass of the cell required just to grow 1 gram. So the same situation was true for glutamine, no maintenance coefficient. And we'll talk more about that. But the lactate production rate or yield, as I said, theoretically should be constant, but it's not. And I won't go through each of these points because we don't have the time. But if you look at the data on the right, the average across all the cell lines that we looked at is approximately 1.5. And as I said, the maximum is 2. So that means that glucose is inefficiently being used. Only 25% of it is being fully oxidized. And so a lot of the energy is being wasted. And that is evidence of the Warburg effect, and we'll say a little bit more about that in a little bit. What makes this situation even more complicated is that the residual concentrations of glucose and glutamine also affect the rates at which they're taken up by the cells. And you may know that these are partially substitutable energy substrates. That's unique about animal cells, that they're using both. And you see there's a lot of scatter here. And the reason for that is that most of the studies did not control for the second substrate. In other words, if one becomes limiting and then the other one becomes limiting, you're going to get some skewed data. Was one study that we found that was well done by Acosta et al. And those are the curves shown, excuse me, to the left and to the right for glucose and glutamine respectively. And it shows as you reduce the concentration and if you restrict the availability of a given substrate, the yield of that on that substrate jumps up and is almost 2 orders of magnitude difference. So that's quite a difference. And what they did differently is they made sure that the other substrate in those studies was controlled and was not limiting. So this data certainly complicates the picture, but it also points out some opportunities in terms of how substrate should be fed to a cell culture to get the maximum potential yield, which has cost consequences because those are the two substrates that are used to the greatest extent and therefore contribute to the cost of production. So the picture's getting a bit complicated at that point. We weren't quite sure what to do, but let's just take stock of what we have found so far. So we said the classical monode kinetics, does a fairly adequate job of representing the behavior of cells under nutrient restricted conditions. We did point out the presence of serum may modify the sensitivity of cells, and so that does need some more investigation. The studies done for inhibitors, also those models seem to work fairly well there, but as we said, they tend to overestimate the inhibitory effects in the way that those experiments are executed. So, but I think if we had better data there, we could model those parts. The more problematic part is the big variability in the biomass yield coefficients, the high degree of lactate production, and that points again to the Warburg effect, which is something that has been perplexing scientists for the last 100 years since it's been discovered. More is starting to be known about it, but essentially animal cells are very inefficient in terms of how they use the substrates. And that those classic PERT kinetics do not adequately describe the consumption of glucose and glutamine. And therefore, if we can't predict those, then it's very hard also to predict the accumulation of the waste metabolites. So now what? Well, we found that we had to go one level deeper, and that is to look at what's happening inside of the cell. And fortunately, there were a few studies that were done that looked at the ATP generation rate along with the consumption of the substrates that were consumed. And because ATP is essentially an internal substrate, you can apply the same PERT model to that and derive the same parameters. So we did that to the 3 data sets that we could find. And lo and behold, we got very similar slopes. So quite excited to see that. Not that that's a new discovery, but it kind of confirmed the fact that cells of very different types all had the same requirement for energy under normal circumstances. And so when you convert those slopes to a yield factor, you get about 32 gram dry cell weight per mole of ATP. And if you flip that, you can get essentially the feed conversion ratio for ATP. 31 millimole ATP per gram dry cell weight is consistent. For the maintenance term, which is the y-intercept, a little bit more variability there, but they're certainly not zero, and they're not all that far apart. So we took an average. The average is about 47 between these 3 data sets, which is interesting because if you take those numbers and you plug them into the equation above, you can calculate what is the growth associated requirement and what is the non-growth associated requirement. And assuming a 24-hour doubling time, which is 0.693 inverse days, a specific growth rate, you get 22 millimoles required per gram dry cell per day. And for the maintenance, it's 47 for a total of about 69. But what's interesting there is that the maintenance term is higher than the actual growth requirement. So that's another indication that the cells are quite inefficient in using substrate. And a lot of what you're feeding them is actually going to maintenance and not even to producing growth. So what does that all mean from the modeling standpoint? And we don't have time to go into this, but we started looking at literature for models that would be would account for the energetics in a cell, and we frankly did not find very many. And then I remembered that a co-student of mine at Tufts University were getting her PhDs at the same time, Don DeMasi, had published a thesis on animal cell metabolism and modeling, and I pulled that out and looked at it, and pretty impressive work, and it seemed to fit exactly what we are looking for, and that is something that's a simplified model that also is predictive of various different conditions. So I got to give a shout out to Don. He may even be on the call today. But there's more explained in the paper, but even there we don't go into the full detail. Uh, it's still fairly complex, but not as complex as other models. As we said at the beginning, we need something that's a bit simpler that we could put into an engineering model with computational fluid dynamics that would then allow us to predict the performance of a reactor. So I'm going to stop there. We'll move on to the conclusions. I'll let Elliot finish it up, and we'd be pleased to answer some questions afterwards. Thanks.
All righty. Yeah, thanks, Bert. Not the easiest content to present, but hopefully that gives folks a flavor of some of the analysis that was done in the report. Really a sort of first effort, I'd say, of pulling that data from papers and converting it into the units that are going to be most useful for people. So in terms of conclusions and recommendations, future directions, one of the main conclusions, as Bert was mentioning, is that, this is not necessarily revolutionary or new, but wild type animal cells probably not the best suited for commercial production of cultivated meat. The reason being that a lot of the energy is poorly utilized. It goes to maintenance rather than growth. And especially with respect to some of the key substrates like glucose, a lot of that is being, again, poorly utilized due to this Warburg effect. Now, on the contrary, because this has been known for a while, there are various strategies that different folks have sort of come up with over the years, but it does imply that perhaps some modification or adaptation of the cells is going to be needed, unless we can perhaps encourage, for instance, lactate uptake or use of alternative substrates, which again we sort of cover in the report examples in the literature. There's also other solutions that are beginning to appear, especially we highlight this paper that was recently published at the beginning of the year, which essentially was able to entirely eliminate the Warburg effect without impacting growth rate. And it's interesting in this study that cells are essentially able to rewire their metabolism into a more oxidative phosphorylation state, which is more energy efficient. And so it points again to the adaptability of cells, and it's really just about being able to take advantage and leverage that inherent adaptability to our advantage in cultivated meat. As Bert also mentioned, that the use of these genome scale and metabolic flux models are also going to be quite important for upstream development with respect to cell lines and media. However, if you were to couple these to this bioreactor physical environment, it's likely too computationally complex. And so we view these as important sort of components of the developmental work. However, it's a sort of separate entity. Ideally, you have cells that are fairly optimized in media that's fairly optimized when you're running these experiments to gather the data that will feed into the models that we're discussing here. Another main conclusion of the report is that this energetically structured model could be best for managing complexity. We know that metabolism is very complex. There's a lot of control points and interactions. The yields can vary by over a magnitude, as we discussed. However, ATP-based yields are remarkably similar amongst cell types, and this sort of points to ATP being a sort of internal unifying currency that might be useful to model on. And so this can also perhaps help us understand and control the effects of inhibitory metabolites as well, based on their impact on cell maintenance or energy requirements. As Bert mentioned, we think that a good starting point to begin to construct such a model would be through this paper in the early 90s that really allows you to model these key intracellular energy pools to help sort of predict some of the cellular dynamics and changing conditions and stressors that cells experience during their time in the reactor. However, we talk about in the report how it's possible that not all of this complexity will be managed by such a simple model, and that we may need to add layers onto it in order to predict other sort of interacting effects like prolonged or intermittent exposure to adverse conditions. And so we sort of envision, a sort of iterative approach where you have to really construct these models, test them out in the real world, and then iterate from there. And then finally, the timing seems to be right to do some of this work. This concept of a digital twin has been talked about for many years, and it's gathering a lot of momentum in other sectors like the biopharmaceutical industry. And coupled with the momentum in AI, with computing power, machine learning and other algorithms that can analyze large amounts of data, recognize patterns, perhaps come up with explanations or hypotheses for why certain observations are being made. These all can sort of work together to bring us to this point where perhaps it is possible to really create a model that is truly predictive of something as complex as a cell growing or trillions of cells growing in a bioreactor. Now, the alternative path is really that with cultivated meat bioprocesses, there's a lot of novelty to this, a lot of first of a kind type of processes that ultimately will have to otherwise be done by trial and error. And so people can debate what's going to be more valuable, practical, useful, whether we proceed and have enough information to proceed by trial and error, or that we put effort into these sort of modeling approaches. I'm not going to make a judgment call on that here, but this is to say that, essentially the timing to do this, the knowledge foundation that we have with respect to the biology seems to be right. So where do we go from here? Well, we have pages and pages in the report of recommendations for modelers and experimentalists. But essentially it amounts to, if you're an experimentalist working in the lab, you can start to generate some of the foundational data and hopefully use this modeling framework to understand, why it is you're generating this data and, taking measurements of certain components. And so again, just a subset of some of the things that are going to be important are cell mass measurements, composition measurements in cultivated meat relevant cells, collecting time series data sets that allow you to track growth, substrate use, metabolite buildup under different time intervals to understand these sort of concepts of maybe cellular adaptation and memory, and then also measuring energy balances that will enable us as sort of like your standard set of parameters that you might be measuring in your process. And that will inform the ability to begin to construct these energetic space models that should help us understand how cells reallocate energy under different stressful or varying conditions within the reactor. We also need to, as Bert mentioned, increase understanding of temperature and pH related effects, which tend to be more global in nature, and begin to essentially construct this cell model that we discussed, couple it with CFD models, and begin to really test it in the real world and iterate from there. Now, enabling comparison between data sets is going to be important. So as we mentioned, using defined media formulations going to be pretty important. That can get rid of a lot of the noise, of serum effects and other effects. We also need, sort of replicable methods across labs, especially when we're taking measurements, that are perhaps hard or difficult to do in some cases, so that we're all speaking the same language. And certainly a lot of this can be accelerated if we can come up with ways to get industry involved, to create incentives to share data or platforms where sharing data is more amenable to industry or private sector. So future directions, as mentioned, we sort of envision this as an open source and collaborative effort. We know there's some efforts going on in the private sector related to this, but really to have an ecosystem-wide impact on the field, ideally this is something that's open source. And so, perhaps you're working on this already, you're interested. We want to know about what you're working on and how it might fit into this overall paradigm. So please do get in touch with us if this is something that you're exploring currently. Also wanted to mention that, from a tooling standpoint, having, again, these cells and defined media will be important. And so we recently announced the acquisition of bovine cell lines that grow in suspension and defined serum-free media. We think that these can serve as a nice standard tool to begin to fill some of the gaps that we discussed in the report. We currently have a wait list for getting access to these cells. And so if you want to fill out the the form that I think yeah, Renee just dropped in the chat. We will get in touch with you as soon as we're ready to start distributing those cells. Lastly, I wanted to end by thanking all the other folks who helped with the report. So Amanda Bess, our analysis program manager, for cleaning the data, revising the report, managing the project, really instrumental in pushing us to the finish line. Our former bioprocess scientist, Faraz, who helped with the literature review and the data analysis. Our technical advisory committee, Casey, Karen, James, Min Liang, who brought expertise in bioprocess and cultivated meat fields for feedback, and also modeling advice from Simon Hubbard and Don Damasi, as mentioned. So thank you for listening. Hopefully that gives you a flavor of what's in this report. We encourage you to read the full thing. And for now, we'll be happy to take any questions you might have. Okay, we'll just start from the top. Bert, I'll read some to you, I guess, as a starting point. How's that sound?
Mm-hmm.
Okay, one of the first questions from Shirley was, how should we approach unknown inhibitory factors? So for instance, if we observe an inhibitory effect of spent media, but not from obvious factors such as lactate and ammonia.
Right. Yeah, no, good question. That data may still be lacking. There was progress made in the BioFarm literature on finding some small molecules that also contributed to inhibition. But I wouldn't dismiss lactate and ammonium. For instance, what hasn't been looked at is the synergistic effect. So what is the effect of both lactate and ammonium together at different pHs, at different osmolalities, depending on how much salt has been added. So there's a lot of permutations there that should be examined to make sure that we've accounted for those. But to find the small molecules, I mean, that's outside of the modeling purview. And that's where tools like metabolomics come in. And I think I can't emphasize more the power of metabolomics. I think for this industry as well would be well served using that, because you can identify pathways that are activated, which then give clues to what kind of toxicity has been experienced by the cell, and then be able to identify those metabolites. And once those, of course, are identified, then of course you do some dose response, similar to the studies that we showed, to develop the kinetics of those. And if there's a way to control the production of that inhibitor, obviously something should be considered from the cell line development standpoint. So I don't know if that answers the question completely, but it's best I can do.
Yeah, I guess I would just mention that the model essentially accounts for allowing other variables within it. And so if you do discover an important additional inhibitory factor that you want to model, then you can include that as a sort of additional factor that you're looking at. But yeah, you'd want to be able to map it out in terms of its effects so that you can actually fit something to it.
Right. Yeah, no, that's a good point. Remember, I pointed out individual factors for each of the inhibitors. So you just simply add one to it to account for that other inhibitor.
Yeah, I think next question is also for you, Bert, and I think this is something that I asked you at some point along the way, but is there a way to model or account for growth factor signal transduction kinetics? How are we looking at the sort of assumptions when it comes to the media composition?
Yeah, we didn't really talk about that. I made mention that there are other types of models out there, such as genome scale models, flux analyses, and things like that, which are more complex. But I do think they have their place in cultivated meat in terms of laying out some details of what goes on in a medium. The model that we're proposing really assumes that the basal medium has already been identified, and that you can then go about determining, growth rates, inhibitory factors, substrate depletion based on that basal medium. So this model is not proposed to help design those media, from scratch. I mean, we're talking 50, 60, 70 components possibly. The same for cell line development, right? What pathway should be targeted? How would you modify it to get better performance? That is outside of the purview of this particular model. But it kind of gives us clues in terms of what are the most limiting phenomena in a bioreactor, which we then can turn around and say, well, if we were to remove this particular constraint, what would the effect be on overall performance? And therefore, we can then prioritize what things we may want to look at. The growth factor story is a complicated one. I believe in the long run, you won't even need growth factors. So that may be heretical, but that's my prediction.
Yeah, I mean, some of that's actually already bearing fruit. I mean, most of the more recent FDA approvals for products, if you actually look into it, most of these products are using less than 2 growth factors, or in some cases actually 0 in their media. And so there's definitely a trend to creating, sort of protein-free solutions in the cultivated meat sector, as has been done in the pharmaceutical sector as well in the past. Next question is for me. What are the glucose concentrations in conventional media like DMEM, RPMI, E8, E8, so on? Off the top of my head, I think they're typically between 3 and 5 grams per liter. But I guess the important thing to realize is that during bioreactor operation, you're not necessarily this concentration might not be what is in the media, because you might be giving concentrated feeds at certain time points in the process of these key substrates like glutamine, like glucose, etc. Which is why, from a modeling perspective, if you're able to predict that in terms of what the cell needs, then you can hopefully use less or fewer quantities of glucose and glutamine and other key amino acids, while maximizing their yields on biomass. Was that explained right, Bert? I mean, that's sort of the idea here is how to really leverage the model so that we're not using so much glucose and glutamine.
No, exactly. And that's the principle behind fed batch cell culture, right? Is that typically you're feeding in a concentrate of glucose at the rate at which it's being consumed by the cells. So the residual concentration in the reactor remains very low. If you just do a batch culture with 3 to 5 grams glucose, and you start at the beginning, the cells are going to use that very inefficiently, and you're going to build up a lot of lactic acid. So that's the reason why that is done. And I would go one step further to say that continuous culture is going to be even better at handling that sort of thing, because once you're at a steady state concentration is really what the cells are going to see. So anyway, it does have a lot of implications.
Yeah, good question. Other question for you, Bert. So someone is asking, I've worked on developing CFD models for large-scale bioreactors. With my colleagues, we've looked at cultivated meat, but didn't progress much due to a lack of suitable kinetics. Do you think with this report and current work, suitable kinetics exist? Similarly, in developing these CFD and kinetic models, a major challenge is validation data. So to what extent do you think robust data are available?
No, that's exactly the point, right? And that's why we undertook this work, because there are no good kinetics that have been published that you can simply stick into a model. And the work to be done is to couple CFD with a biological model, as we pointed out, that is not overly complex, but still is predictive. And that, as far as I know, just doesn't exist. And so that needs to be developed. We are recommending the Damaski and Swartz model as a good starting point for going forward, for collecting the data and fitting it to that model and see how well it works. As Elliot mentioned, we may need to tweak it. We may need to add a few other metabolites to make it more complete. But that is work in progress, and I think that's precisely where we need to go.
Yeah, exactly. Another question is there something I could do to help as a CFD engineer, or are processes limited by the biology side?
Yeah, and that's a great question again, right? It goes right to the point. The short answer is we don't know, right? We don't know if bioreactors are really more limited in terms of physical phenomenon or biology or some combination of the two. And that is why we're proposing to couple a bioreactor environment model, which is probably CFD to represent the three-dimensional aspects of the reactor, with the biology model to see if the two of them together can predict performance. The only point about validation is, yes, absolutely, these models need to be validated with data, but it's going to be an iterative process because nobody's going to build a large-scale reactor and then hope it works, right? Step up, building prototypes, using scaled-down models, and then being able to validate these models, and we kind of inch our way up the scale. Did I answer that question? I think so.
Yeah, I certainly don't have an answer. I'm not too much of an expert in on the CFD side of things, so I will take that as
Yeah, I
it.
was going to say that something that can be worked on immediately is how do you couple the biological model to a CFD model? So even if the biological model isn't perfect, at least we can get the mechanics worked out of how that is to be done, and then we can start tweaking the biological model. We can see what is the computational power required to run those models, and if it's even feasibly possible. So there is work that could be done. Sorry.
Good point. And I think also like, yeah, determining the time scale at which you're looking at will be important as well from the computational side of things, and how much resolution you really need and what are the trade-offs there. All right, there's a comment from Peter about there's some interesting work done on the modeling of excess kinetic metabolism by Zhang in 1995, including the observation that waste metabolite production is largely dependent on excess concentrations of glucose and glutamine. Have you considered putting forward these kinetic descriptions in the report?
Yeah, the answer is yes. I am aware of that study, and we do talk about it in the publication. I looked at that, and I would say they actually had the simulation results were highly predictive. So the model worked well. My issue with it is that it was very counterintuitive, right? This excess concentration is a theoretical construct, and it kind of bypasses the whole energetics part of the description. And in my opinion, that the underlying energetics is very much how a cell manages its metabolism, particularly the catabolic pathways, are very much controlled by the availability of ATP and NADH. And the Zhang model sidesteps that whole issue. And so my concern is that if you have to augment the model or add to it, then you start almost from scratch. Whereas if you have a model that's more realistic in terms of representing the pathways, adding another amino acid, an alternative substrate, mechanistically should be easier to do in a model such as the one proposed by Damasi. It was a long-winded answer.
Yeah, thanks. I'll mention that hopefully people, if they dive into the report, you'll notice that we probably covered every single foundational paper from the late 80s through the early 2000s and to the present day that we think we hit on and looked at. And so, but if there are things that you find that, you know, we might have overlooked in the literature review, definitely reach out and let us know. But we think we've been pretty comprehensive in looking at a lot of the key papers that have been published over the years. This is a question that I can take. What are the impact on regulation using GM cells? So yeah, I assume this question comes because we mentioned you might want to modify cells in order to make them improved. So yeah, obviously, you know, genetically modifying cells would add a layer of you know, regulatory scrutiny. Some regions it's more or less accepted by consumers, and this is also a ever evolving sort of state of affairs when it comes to food production in general. With that said, you know, there has been a product in the US that used genetic engineering. Upside Foods product was, you know, had that implemented for their immortalization process, and that some forms of gene editing in a lot of updated regulatory frameworks are sort of viewed differently, especially if you're not using transgenes. And so it may become quite regulatorily speaking permissible to do editing in the way that, for example, we highlighted, where you knock out some endogenous genes related to lactate production pathway, and are able to produce, essentially very high performing cell lines. But also, as mentioned, what typically happens is that cells can adapt quite a bit as well in culture, and being able to select those through just sort of typical experimental pipelines can also go a long way and avoid the modification sort of scrutiny. And so, this is a consideration that companies are going to have to make. We surveyed the industry back in 2023 and found that I think somewhere around maybe 60% of companies were exploring gene editing or engineering from an R&D perspective, and a subset of those from a commercial perspective as well. Maybe around a quarter of companies. But probably a lot of the first generation products are going to be non-GM. But we'll see how that evolves over time. I don't know if you have anything else to add, Bert.
Well, it'd be interesting to see how the economics works out for anybody using wild type animal cells. I mean, it suggests that the economics are not going to work out based on what we've been finding. But there are various different product designs. There are hybrid products. So, I think that verdict is still out. But my prediction is in the long run that we're going to be using genetically modified cell lines to achieve the maximum performance and therefore the lowest possible cost. So that's going to take some time, obviously.
All right. Next question is, did we consider any work using microcarriers? How might that impact results?
Yeah, interesting question. Yeah, no, I guess it's my bias somewhat, because as a consultant, I've done a number of evaluations, which points to the fact that microcarriers is probably not going to be economically viable. Now, it depends what we're talking about. There's a proliferation stage typically, and if your differentiation stage, there could be a second stage where you seed cells onto some sort of scaffold to differentiate the cells. Could be done on microcarriers, but then you're going to have small pieces. More likely, you're going to seed it into some sort of porous scaffold to form some semi-rigid tissue as your product. So the short answer is no, we didn't specifically model cells on microcarriers. We did point out in the study that there's very little data information on cell differentiation in general, and how they gain mass and how they shift in composition over time. We included one example report and sort of fit a kinetics or empirical model to it, but certainly not structured. So there's still a lot of work to be done in applying these models to stationary culture where the cells are attached to some surface.
Yeah, I'll just drop on the chat too, just off the top of my head. There are some people that have looked into this a little bit on the modeling side of cells on microcarriers, some recent literature. So, it's out there. Some people are exploring how to sort of look at microcarrier growth, and whether or not that I haven't seen any real sort of techno-economic evaluations, public ones at least, that compare and contrast microcarrier scenarios versus suspension scenarios. All right, some more questions rolling in. Bert, I think I'll pass this one to you. So what do you think about how to apply the gradients and spatial distribution of the fluid environment inside the reactors with the local heterogeneity of cellular growth behavior?
If I understand the question correctly, I mean, that's sort of the essence of what we're trying to do, combining a physical model of the reactor, which is probably going to require computational fluid dynamics that breaks up the environment into these elements, fluid elements, right? And using mass transport and momentum transport equations between the elements, you can kind of predict how these gradients propagate throughout the bioreactor, right? And that can apply to concentration gradients, to shear gradients, uh, temperature, pH. Oxygen concentration is sure to be a factor because of its low solubility. So once you've set up the three-dimensional aspect and those physical components, you can then layer into that the models. And as I said early on, that you're going to have to execute the cell model for every one of those little fluid elements where the fluid element represents a difference in conditions. And then it's a matter of putting that all together to estimate the overall performance or overall productivity of the bioreactor. You can envision, some cells are not getting enough oxygen, maybe some are getting too much. Most of them are getting the right amount. So, but then what does that mean in terms of the overall reactor? Something we didn't touch upon is the memory effect. We do talk about it in the paper. There's very little information and data available on that. But if you remember that diagram I had at the beginning where the cell is going through these different zones in a reactor. Now, what if it's starved for a second of oxygen, right? And then it gets maybe a glut of something else. What is the impact on the cell over time? And so there's probably some sort of memory effect that needs to be accounted for in future models that is a whole nother dimension that needs to be examined.
There's a follow-up question, still related to the microcarriers, about economic feasibility, even if the microcarriers are edible, so that they're basically considered maybe part of the end product. So you're harvesting the cells with the microcarrier, which I know some people are looking into. I think it would really come down to an analysis of, you're looking at the bioreactor volumetric productivity, and how much mass of cells can you generate on a microcarrier, versus in suspension, and whether that delta is sufficiently covered by the microcarrier fraction itself. I would assume you'd want to really look into that.
Yeah, I've looked at those scenarios too, and it's also difficult to make it work out because the real estate on a microcarrier is not that great because you're restricted to the two-dimensional, unless you have a very, very small particle size, which is difficult to achieve. But I'm happy to follow up with you on that later if you like.
All right. Well, there's still 70 soldiers who stuck with us the whole time, but I don't see any other questions. If not, we can close out. Thank you everyone for attending and listening and being interested in this. Again, if you're working on this, let us know what you're working on, and maybe clear a week on your calendar to read the full report, and get a sense of what's really in there. So thanks again for attending.
All right. Thank you all. Bye-bye.